From the Chronicle Podcast Network, this is the Skin Spectrum Podcast, your weekly conversation about ethnodermatology. This discussion is all about our skin, the doctors who treat it, and the diverse patients who live their lives under it. On today's Skin Spectrum Podcast, we continue our summer series of weekly reports with a special focus on the Black dermatology patient. Our second episode of the Skin Spectrum Podcast Summer of Dialogue focuses on Stevens-Johnson syndrome. Our guests for today are Dr. Neil Shear, previous head of the dermatology department at Sunnybrook Health Sciences Centre in Toronto and professor emeritus at University of Toronto, and Bruce Carleton, senior clinician scientist at BC Children's Hospital Research Institute and professor at University of British Columbia. Here's their discussion.
I'd like to welcome everyone who's listening to this recording. I'm Neil Shear. I'm a professor emeritus of clinical pharmacology and dermatology at the University of Toronto. Much of my career has been in very severe drug-induced diseases, but also especially the very dramatic and horrible disease that's been called Stevens-Johnson syndrome or toxic epidermal necrolysis. This involves extensive blistering of the skin and a high risk of death and long-term suffering. And there's been so much work that's been going on since I started in this field. I am very, very excited and happy that Bruce Carleton, who's a clinician scientist at the University of British Columbia, who's done amazing work in almost all aspects of drug-induced diseases, the genetics, the clinical diagnosis, other investigations, and he's online with me now, and we're going to be talking a bit about what he's done with a focus on skin of color. So just starting off with you and what you're doing, can you explain to the listeners about the work that you're doing and also a little bit of emphasis on skin of color?
This is a topic that's one that I guess I fell into. I'm not a dermatologist, so in some ways I learned about dermatology through the patients that I see with these tragic drug reactions. And once you begin working with these patients, you understand them in ways that you didn't before. And that's one of the things that I have to thank you about for exposing me to this area and to understand them. The damage to the skin and the chronic scarring that occurs causes a lot of concern to these patients and lifelong concern. And one of the first things that I began thinking about, being that I've spent my life mostly in pediatric diseases, is how similar we all are between adults and children when it comes to these kinds of tragic reactions. I work with a lot of children with cancer, and if you ask oncologists, pediatric oncologists, what are the most important adverse events of chemotherapy that they're concerned about, there are things like liver disease and kidney disease and secondary malignancies that can occur in these patients. But if you ask the patients what matters to them, children, adolescents, what matters to them most in terms of adverse effect from their cancer treatment, they talk about the loss of hair, their body image, and so forth. And I think so much of who we are as human beings is tied to our appearance, not just vanity, but just in general, the way we interact with people, our smiles, the way we look at each other. This is one thing that compelled me to work in this particular area, was an understanding that these patients really suffer a lot from not only a reaction that occurred and then has resolved, but is ongoing in terms of having to manage this on a daily basis the rest of their lives. And that's really how I came into this particular area.
And when we were looking into this area, obviously a lot of your emphasis in the beginning was certainly on genetics, and you've built an incredible center doing all this cutting edge genetic work. Can you explain how that fits into what people should know about?
There are 4 key steps in drug biotransformation in the body. Drugs are absorbed, they're distributed, they're metabolized, and they're excreted. 4 steps: absorption, distribution, metabolism, and excretion. And in those 4 steps, they're controlled by genes. And so one of the things that I began thinking about is variability between patients in these 4 steps that occur, and how could we actually look at that and examine that? And in the early 2000s, the cost of genetic sequencing, the ability to actually read the genetic alphabet in individual patients, became much more affordable. In the early 2000s, In fact, in 2002, then U.S. President Clinton, with Francis Collins at the U.S. NIH, announced the human genome had been sequenced. They looked at 1 million variants throughout the human genome. It took them 13 years and $2.7 billion to do that. Today, I can do the exact same thing in about 10 hours for $50. And so that technology, that massive boost in technology that allows us to look at the genetics, made it possible for us to look at those 4 steps of biotransformation from the genetic perspective and how patients differ from each other and how these steps might in fact relate to the development of conditions like Stevens-Johnson syndrome and toxic epidermal necrolysis. And since that time, there have been a number of very robust genetic traits that have been found to be strongly associated with the development of these reactions. And so we're off and running now with understanding a few markers, about 10 that are strongly associated with these conditions. There are more to come. And I began looking at that region of the genome to be able to understand its utility in helping us to predict in whom these reactions might occur before they got the drug, and also to help us understand in the patients who actually had these tragic reactions if there was a genetic basis for their reaction.
There's 2 outcomes, I guess, or points I'd like to ask you about then, because you've laid out a pretty nice story of all those steps that might have to go through with drug metabolism and all these effects that can happen. But if we're looking at things like not just people's genetics, but their racial type and skin color, and we see them beforehand, they're going to get a drug that might cause this disease. Can genetic testing protect them? And I guess part 2 is, if genetic testing wasn't done to protect them, if they do have a reaction or a relative does, can genetic testing help guide a safer future and a safer use of these drugs?
This is exactly where we need to go. So yes, genetic testing can help to protect them. I'm thinking now of an elderly patient of mine who is in her 80s, who was diagnosed with gout, was put on a medication for gout called allopurinol, and ended up with Stevens-Johnson syndrome. She has the genetic marker that we know puts patients at increased risk of this particular condition, increases their risk by 14-fold up to almost 600-fold. Depends upon the study that you look at, but between 14 times elevated risk all the way up to almost 600. And she was positive for that particular marker. And so had we done this very inexpensive test in advance, we could have avoided the tragedy that's happened. She unfortunately has had a condition that's much more difficult to control and has received multiple immunosuppressive therapies, multiple hospitalizations. And we finally now, after several months, to get her skin disease under control, but it's been quite a lot of suffering for her and her family. That gets to the second question is, can we make drugs safer in the future, for example, for family members? Because as you can imagine, her children who are adults come to me to say, well, if my mom was at risk, are we at risk? And how would we be able to determine that? And because we don't want this ever to happen to us, because they've watched their mother suffer from this. And so that's definitely something that we can offer, is that particular testing. And although the markers are great, we have 10 of them, they're not sufficient to explain all cases. And so we continue to look for others and use other tests to understand which drugs are actually responsible for these reactions, because they mostly are, in fact, drug-induced reactions, as you know.
These reactions, now drugs generally go through a very comprehensive review by Health Canada, FDA in the United States, and across the world. And, if you see severe reactions, often drugs never make it to market. But we're talking about drugs that have somehow got to market, and because it's a rare event, may not have shown up in these trials. So we need to really go poking around to find out what's going on and try and shine a light. And it sounds like that's what you're doing. You're really shining a light in an area that seems to be so mysterious about all these steps, but it isn't once you get down to the crunch, at least to a practical level. But I do want to get more into the brown and black skin and where people are from. And does it matter if somebody's mother happened to be from South Asia or other examples?
Yeah, I think the skin color is important. So some of these drugs, first of all, these are rare reactions, but rarity means nothing if it happens to you or it happens to a family member. That's one of the challenges, is that People have said, oh, they're rare. So how are we going to be able to study something that's so rare? Well, we form global networks because we are one race of people. We are a human race, and we work across boundaries that mankind has created to find solutions that impact us all in that single race that we're part of. But within that single race, we of course have many variations, and those variations are important in terms of the risk for these reactions. And, our good friend, Dr. Wenhong Chung from Taipei, was the one who found that the commonly used anti-epileptic medication for seizure control called carbamazepine or Tegretol had a genetic variant associated with its risk of causing Stevens-Johnson syndrome in our Southeast Asian brothers and sisters. And that risk was elevated by more than 2,500 times. And that became a very powerful article in the journal Nature that caused our Asian countries to evaluate how they are providing these medications, given that those genetic traits appear more often in Asian populations. And then I think another important point that's often missed here is that for patients with skin of color, many of these drugs were approved in the 1960s and 70s from very large countries like the United States and Canada. And we are, in fact, different genetically than we were in those previous decades. We have larger numbers of immigrants from other countries now than we had before. And so the makeup of patients that were originally used to support the licensing of a product has changed. And consequently, as immigrants from these countries move here, they marry others, and the ancestral diversity increases even further. And so this is one of the reasons why I think it's really important to focus on skin of color, because they tend to be underrepresented in these drug trials in past decades.
Just a straight on question. How is Canada doing in terms of genetic testing of people? You did mention that it's being done in other places, but how is Canada doing in genetic testing of patients? And where is that going to be going?
To be honest and to be transparent to our listeners, we're doing terribly. We're doing poorly. We have not recognized the importance of these markers. And I brought up one example of a patient. I've got many others of patients who would have benefited from such testing being widely available before prescribing occurs. And if you're thinking about these things from the standpoint of how much does it cost to offer a test to large numbers of patients when only a few patients will test positive, if that's the economic argument you're making, then these tests are never going to be cost effective to provide. If, however, your analysis is the avoidance of suffering, the avoidance of hospitalization, the avoidance of post-traumatic stress disorder, which occurs in these patients with any future medication they take for fear that they might have a similar reaction, these become very cost-effective things to offer. I have some trouble in this country of less than 40 million people that we spend about $35 billion a year on drugs, but we can't afford necessarily, or we haven't decided to afford or make a priority genetic testing to ensure safety where we know that these robust markers exist to prevent harm. That's, I think, the critical thing. And there are many cases of this. I get asked all the time as somebody who actively does this type of testing for individual patient testing. And that's because clinicians want access to this. The patients want access to this. But at this point, federally and provincially, I'm not hearing really any call for managing this by offering testing that we know is helpful to these patients.
It does sound like it's something that should be part of the whole flow. And we published a paper the past year or two about a survey we did in Toronto just to see what clinicians in internal medicine, et cetera, knew about screening. Actually, it was an educational survey because we would answer after they gave their opinion what is known. And it really is not, I think people respect it when they hear about it, but it's just not part of what is being taught that I can see. This is something that has to be embraced by the government to make it happen. And it's in all people, but especially in people with skin of color. I think there's a higher risk, and depending on where your family is from, you can pretty well know which genes you need to look for. And you can know the results very quickly. What you've done with bringing down the cost is spectacular in terms of the impact of that. It'll save lives. So I think there's a lot to be done. So where do you think you're going now? What's exciting you about what's coming up?
Well, I think that the way to manage this is not for me as somebody who studies the pharmacogenomics, as we call it, of drug reactions, to go to government and say, hey, you ought to support paying for pharmacogenomic tests. I mean, I can walk in the door and they'll know that that's my bias coming in. But there is a group of people that can advocate for this much more impressively than I can, and that's the patients that we serve. And so my goal is to offer as much free testing and availability as possible to generate the evidence, to get that army of humanity that I know is out there, of these patients who benefit and their family members, to say to government, this is enough. We need this paid for, and we're willing to support this, and we want to support it. The costs are fairly small when we scale these things up. So just to give everyone an idea, we often look at gene variants. Each variant that we test is so much less than a penny to test. I can't even tell you what portion of a penny it is. But the problem is I can't buy the reagents and the equipment and the laboratory space and the technicians and the highly qualified personnel just to do one test at a time. So the more tests we do, the price comes down. It's like any other technology, iPhones, cars, anything else. As we scale up the number of items that are available that people purchase, the price overall comes down. And so that's one of the things that's going to be critical here, is that we understand that we've got to get collectively together to do these things, to get commercial companies that are laboratory testing companies interested in investing in the technology and the capability and the personnel to make these tests widely available. And they understand that too. It's a numbers game. If we're testing, 10 patients a month, it's always going to be cost ineffective. If we're testing thousands of patients a month, then it will be cost effective, and there will be commercial providers who will want to provide that type of testing. And individually, the testing is actually pretty inexpensive. I think the other thing, Dr. Shear, that I wanted to mention is that we often think about the number of positive tests compared to the number of tests overall we had to run to get that positive test. And I would say that that is an incomplete assessment of the value of testing. Where is the extra value? The extra value is in knowing that you are negative for a variant that increases your risk. There is value in a negative test. It's not just simply, well, you didn't test positive, therefore your test was of no value. Ridiculous. Any of us that would test negative would say, whew, that's good to know. I'm glad I'm negative, right? And so part of the problem that we've had with moving forward on these tests is people understanding that the value of the test should be in both positive test results and negative test results, frankly, for patients that we serve.
Really, really good, strong, compelling point. And we really need to do that. There's just so much that can be done, and we've done so well. And what you built up is a good example of it. You've got a network of people that you're working with basically globally, which is amazing and really great. Lets us understand what's going on in so many different populations and healthcare systems, but certainly a lot we can do in Canada. We really need to do a lot more. It's disappointing to see that we haven't done that. One of the strengths of the Stevens Johnson Foundation that Sonia Whyte-Croasdaile is the president of, is they're really out there pushing this story and getting the information out. And in working with them, as you and I have, patients and survivors tell their stories is really, really compelling, but also how others who hear those stories say, yes, that makes sense. That's what happened to me. The support is necessary, and that's a big part of this, but the genetics is something that can really change the landscape. It can really shine a light in a dark place and just make everything start to make sense. And I think we're really on the verge of that. Do you feel that we're on the verge of something that, we're really going to be making a difference? We'll have a very different conversation in a couple of years?
Yeah, and I think in many ways we already are. I can think of one TEN patient in particular who we did genetic testing on. Unfortunately, the medications that she was on at the time when she developed TEN are not on this list of 10 HLA markers that we can use to guide what exactly occurred. And so we've been developing other assays, other things that we can do to understand which of the many antibiotics that you are on is actually likely the causative agent to your skin condition. And we've been using the patient's lymphocytes, their white blood cells, in a sense, to be able to understand in the laboratory environment which drugs that they are susceptible to. And that's been extremely helpful for patients who want to understand the causative agent. And then secondly, if I am allergic to this product, if I cannot use this product in the future, then what's the alternative? And am I allergic to that? Because we all know that these patients suffer tremendously from having had these reactions and are worried about other medications. So we can actually pre-test those other medications now using their blood cells and determine if those are safe to use as well. And we're beginning to triangulate all of these research tools into a clinical practice standard that I believe in the next few years will result in safer drug therapy for all of these patients and helping them in the future as well, not just knowing what their current culprit agent is, but also what they can safely use and not use in the future.
Yeah, thanks, Bruce. I think, and also for listeners, just to say, we're not here to push drug use. We're not saying that. But we are saying that if people do need a drug and they do need a medication, we can dramatically enhance the safety for so many of these and prevent these very severe reactions occurring by doing this kind of testing and understanding the genetics more. That's, for all patients. Anything else, Bruce? Any last minute comment you want to add?
Another area that is currently being looked at by the U.S. Centers for Disease Control and my lab is, what about these types of skin reactions to the COVID vaccine? We are seeing some reactions to the COVID vaccine, and we're also looking at the genetics of vaccine reactions. So it works for drugs. I believe it will also work for vaccines, and we're really excited about that as well, because as we roll these vaccines out and we understand in whom adverse reactions occur, we need to understand the biology that underlies the adverse event so that we can avoid those in the future. And that's really what this work allows us to do, is not just predict and prevent, but also to understand the biology of the adverse event in a new way for the first time. That's been really exciting.
Oh yeah, that's a brilliant end. Thank you so much for that, because it is something we're facing all the time. Thank you very much for being part of this interview and for all the great work you're doing and sharing your knowledge and experience with us.
Thank you very much.
Now we have a very special guest, and it's Sonia Whyte-Croasdaile, president and founder of SJS Canada. Very happy, Sonia. Thank you for being here. And the work that you're involved with too, in terms of getting better genetic testing and screening. We know that some of these reactions are genetically determined. Screening does take place in many parts of the world. And much of that is based on skin of color. So people with darker skin, with Asian skin, black skin may be at more risk. Most of the information is with the Asians, but there's more information coming. What's your feeling about what this gene testing might do if we could only get that going?
If that could get going, that would be an amazing thing, because it would help people like myself and others to realize before it happened things that they are susceptible to. And so it would make life a lot much easier, especially in my case, being someone skin of color. It was very difficult to see all the sores coming up on my body because my skin was so dark, so you could hardly see it. It wasn't until those became like blisters that most of them were visible. They were all over my body. So with the genetic testing coming up, it's kind of making arm us before we go out in the SJS battlefield, pretty much. So we'll know if we carry that genes. And with the research and everything that's going on, you will be advised and supported on what you should stay away from, and what you need to be cautious about. So that would be a wave of change within the world and individuals who have these genes.
Thank you. I would say that also Canadians, in terms of the genetic testing, we have Professor Carleton, and then we have Canadians elsewhere in the world who are also very much involved in the testing, like Dr. Phillips. Sonia, thank you so much for coming on.
Thank you so much for having me. My pleasure.
Thank you to Dr. Shear, Professor Bruce Carleton, and Sonia Whyte-Croasdaile for sharing their knowledge and expertise with us today. Join us on August 21st, 2021 for the conclusion of the Summer of Dialogue series, our live colloquium on black skin. The Skin Spectrum podcast is produced from studios in Buffalo, New York and Toronto, Canada by Chronicle Companies, publishers of the Chronicle of Skin and Allergy and organizers of the Skin Spectrum Summit. Visit us at www.skinspectrum.ca or www.derm.city, where dermatology lives. We would like to thank our in-kind partners. At the Nurse Practitioners Association of Ontario, the Canadian Association of Aesthetic Medicine, and the Primary Care Dermatology Society of Canada. Subscribe to the Skin Spectrum podcast at Apple iTunes, Google Podcasts, Stitcher, Spotify, or wherever you get your podcasts. This episode was produced by Jeremy Visser with assistance by Arya Impakaris. Research was done by Kylie Rabernick and Catherine Brenders. Until next time, thanks for listening. We'll see you again next week.